activated. What you can see is that when we adenovirally express constitutively active AKT2, we completely rescue AKT signaling in the knockout hepatocytes. So we then asked, does this also rescue the defects in activation of SREBP and lipogenesis? What you can see is that I showed you before, knockout hepatocytes have a defect in insulin's ability to activate SREBP, lipogenesis, and its targets. However, when we constitutively activate AKT, we now rescue the induction of SREBP and lipogenesis, demonstrating that attenuation of AKT is responsible for the defect in the induction of SREBP and lipogenesis in these knockout hepatocytes. So it seems that while AKT regulation of mTORC1 is required for induction, it's not the only pathway, and there's another pathway downstream of AKT that must function in parallel to mTORC1 to get full activation of SREBP and lipogenesis. I hypothesized that this would be a liver-specific substrate because in other cell types and settings, we've seen that mTORC1 activation alone was sufficient to get activation of SREBP. When I was studying for my PQE, I knew I would be asked this question and was trying to understand the difference between other cell settings in the liver. And I came across a paper from Brian Goldstein's lab that identified a liver-specific isoform of these ER retention proteins, the INSIG family. This liver-specific transcript is known as INSIG2A. Essentially, this is downregulated by insulin, and the authors hypothesized and it's believed to enable SREBP to be activated in the presence of insulin, even if there's a lot of cholesterol available. So the cholesterol mechanism does not override the insulin signal when cells want to store and produce lipid. This presumably would act upstream in the functional pathway for SREBP activation to whatever mTORC1 does downstream of SREBP, downstream of processing of SREBP. So the first thing we wanted to know was whether or not AKT is involved in the regulation of INSIG2A. As I mentioned, insulin is a stimulation that actually brings down levels of INSIG2A, so we use two different paradigms. One is in primary hepatocytes where we can treat with insulin, and the other is this fasting and refeeding model that produces insulin production endogenously. They really look very similar, so we can focus our attention to the in vivo data on the left. What you can see is that expression of INSIG2A is suppressed, as was published previously, in the livers of mice after refeeding. Importantly, this was not affected when you pre-treat mice for 30 minutes with rapamycin. Strikingly, however, if you pre-treat with an AKT inhibitor, you completely reverse the refeeding